Well, thank you very much, Richard, for this very nice introduction, and thank you very much for this invitation. It's a pleasure and it's a big honor for me to be in front of your entomologists of the Royal Society, Royal Entomological Society. That's great. And I think it's a kind of symbolic that this year, I don't know, maybe every year, meanwhile, this talk will be given at Imperial and at Engineering Department. So which means that this lecture fits pretty well to this audience, audience of you and this lecture hall of engineering, because it is a lecture on the borderline between entomology and engineering, if you want. So fly in the ceiling, how insect adhesion research can inspire technology. Yes. Oh, yes. So this is the only one slide with a German typo. Bionic, it's a German word for biomimetics, because biology and technology. And of course, the English word biomimetics or bio-inspired technology. And on this slide, you see the philosophy, looking at the nature, wisdom to promote technology, to develop technological applications. But this example with the shark and with the airplane is, of course, very gigantic kind of structures. But we in Kiel, we are focused more on very tiny little structures. And as you know, insects have a lot of interesting micro and nanostructures. And of course, many of you as systematicists and phylogenists, you use this structure to determine species, to understand their phylogenetic relationship. But of course, this structure evolved in the biological evolution for certain functions. And of course, there are a number of functions. There are little sensors, attachment devices about which we speak today, surfaces which reduce drag, maybe not in these animals, but in some insects it is the case. Optically active surfaces like anti-reflectors in the eyes, wear reduction mechanism in the mouth parts, friction reducing mechanism in the joints, sound generation, respiration, filtering, thermoregulation, coloration pattern, physical colors, self-cleaning surfaces. And many of these, when maybe even majority of these functions, have something to do with the micro and nanostructures, not necessarily the chemistry of surface. And this is the interest in my working group in Kiel. So if we take few just randomly selected examples of micro and nanostructures, what we see here, our database is pretty big, but you see here some structures with the anisotropic friction. So in this case, the friction is higher in one direction than in another one. So there are some structures which reduce turbulences of the wing, optically active structures on the eyes of some dipterans. These structures, they looks like a little Velcros, but this is Velcro holding air bubbles underwater, filtering structures, structures which prevent contamination. It is beetle living under the soil and remain always clean because these hierarchical structures on the covering wings. And we try to understand this principle, and for this purpose, and this is one of the philosophy of biomimetics, in order to mimic something technologically, we should understand how does it work. And for this purpose, we should understand fine structure, we do ultrastructure, we measure material properties, the force range, and we try to not forget what animals do with all that. So we also study the motion associated with these structures. And well, for this purpose, you should also sometimes develop your own methods, because you can measure friction using the big technological machines. There are a lot of commercially available devices, but if you have a fly, how to measure adhesion and friction? So you should build your own kind of technique to do that. To measure hardness, adhesion, friction, and the local and global scale. And if you find interesting properties, we try to mimic that by using collaboration, but also using our own approaches and transfer these solutions into material science. And I'm entomologist, as Richard mentioned, and I will probably for the rest of my life remain entomologist. And for me, the most interesting part of all this business is how these structures evolved in the evolution, how they developed and adapted to optimize the function. And for this kind of purpose, you should do broad and comparative studies. But it has not only biological meaning and not only like academical value, but also if you find out that the same solution for the same problem appeared many times independently in evolution, maybe this solution is the optimal one also to mimic in the engineering. And exactly this solution we're looking for, and exactly this solution are most interesting for biomimetics. And if you speak, we must speak to the people from technology, from the companies, and if you ask them what we would like to do with these kind of devices, what should be the main kind of property of these devices, you will say we would like to have something sticky like a fly on the ceiling. And maybe I would discourage you a little bit because fly doesn't stick too strongly on the ceiling. Actually, the adhesive forces and adhesive strength of the fly on the ceiling is, well, comparable with the typical Scotch tape, but definitely not much better. And that's why if you would like to have something really sticky, you should go on the seashore and maybe look on the barnacles and mussels. They stick really well and even underwater. But if our task would be to use these kind of devices for walking on the wall and the ceiling and to adhere very fast and to adhere to unpredictable surface. Animals doesn't know what would be the next surface they adhere to, and to have something which is not only adhere well, but also fast releasable and also in millions of cycles, and something which adheres to more or less everything, but not to itself. Imagine if the animal would put legs together and they stick like a Scotch tape during the, putting Christmas decoration. This is definitely not good. And actually, it's not a big, challenge to put a big mass on the ceiling. In some American university, people who is promoted, they will be stick to the ceiling using a lot of Scotch tape. And it works, but it is a static situation. It means that this guy can stay forever on the ceiling, but cannot move. And in order to master this situation, it's actually physically, I would say, pretty easy. So you should counterbalance the weight of the animal using a certain amount of adhesion, stickiness to the ceiling, and friction, which prevents sliding along the ceiling. But if you would like to generate dynamic situation, if you would like to walk on the ceiling, then you should solve two problems, and they are not trivial at all. You should make a contact in very fast and reliable way and with a minimal load on the ceiling. We will discuss that later on, why it is important. And you should break the contact also in a fast way and with minimal force expenditure. Imagine if the animal should put a lot of energy to debond from the surface, then it would be probably not supported in the evolution. And of course, in the middle of these cycles, you need to have a strong adhesion. Okay, so if you, maybe majority of you know that insects usually use so-called tripod gait. So they use three legs, two on one body side, the forelegs and hind legs, and the mid leg on the other body side. And this is a very stable situation. That's why the majority of modern robots, biologically inspired robots, they are hexapod, because by this way of walking, you generate a stable gait. Because if I steady in front of you, all my muscles should work to keep the stability. In this case, you can freeze the fly and it's stable because there are 3 points holding stability. But on the ceiling, flies change the gait to the quadrupedal gait, so the forelegs will be in contact. They win exactly 1 point to generate strong adhesion. And this is the case of the fly, but if you take other animals which adhere better than the fly generates something like what we call safety factor, something like 3. It means 3 times own body weight they can keep on the ceiling. Some of them, they can maybe reach 10. But some beetles, they can reach something like about 100 own body weights on the ceiling. So these guys, they can walk on the ceiling using 3-pod gait. They should not change the gait because the adhesion they produce is sufficient to stay on the ceiling. But the animals which produce about 1, like this bug, they are able to use only, they are able to move only 1 leg and use 5 legs to stick on the ceiling, because to have safety factor 1 is very critical. They can fall at every single step. Okay, insects usually are not evolved to walk on the window. They are not evolved to walk on very, very smooth surfaces, but they can do that because some of the plants, they are pretty smooth, but of course, majority of insects, they evolved to walk on the plants, and they have all varieties of surfaces. Some of them are pretty smooth, some of them, they are hairy, like felt-like hairs, covered by particles like pollen grains, covered even by tiny little drops of sticky materials like some trichomes of tomato plants. And the question is there only one type of the adhesive pads insect evolved? Or there are 2 or there are 5? And of course, there are a couple of contact mechanics theory. We will come later to this point. But for us entomologists, maybe the best way to take all these animals to put under the microscope and look on the feet. And that's what we did. And what we find out was initially pretty surprising. We find out that there are only 2 types of pads. The one type, which we call hairy, consists of tiny little hairs. And of course, you can imagine that all these hairs can generate 100% of contact on the flat surface. So which means that the flat surface is not really challenging. And even for the flat surface, you even don't need hairs. Why? Because in order to make a good contact on the flat surface, you need just another flat surface. If you take 2 pieces of glass and put them together, you have a hard time to, well, debond them. They will bond very, very strongly. So, but the majority of surfaces surrounding us and flies, they didn't move usually in the clean rooms. They move in the normal environment, which is dirty, which is rough. In this room, we have the majority of surfaces which are rough. So how to generate good contact on the roughness? The one possibility is to change the smooth contact to very long outgrowths, which can adapt to more or less any surface profile and increase number of contacting points. So, which means that, of course, we reduce the contact area because we have gaps between the hairs, but on the smooth surface. But on any rough surface, we win by the increasing number of contacting points. The same situation we have for so-called smooth pads. So this is the example of the smooth pad, and these pads are indeed smooth, but they are not rigid. They are very soft, I would say like a chewing gum, and they can actually do the same. They can adapt to the surface profile. They made of material which is so soft and can flow into the crevices and adapt also to any surface profile, which means that we can discuss with a physicist among you about contribution of basic physical forces. It is van der Waals interactions, or it is electrostatics, or capillary forces. I would say it doesn't matter. What matters is in the contact mechanics. If you generate proper contact, you generate good adhesion. If you cannot generate proper contact, you would not generate good adhesion. So very simple. So which means if we understand that, how does animal do that, we can also solve another problem, because we know how to generate surface which prevent generation of proper contact. And this is a case of many plants, which on purpose cheat the animals to generate proper contact by having some micro and nanostructures and make surfaces slippery. Okay, so this is pretty old version. I know, well, this kind of phylogenetic tree, they're changing every couple of weeks maybe, but it doesn't matter. The message of this slide is if we plot smooth and hairy pads of insects, we see that both types, they develop independently just among insects at least 3 times, completely independently, which means that these hairs here around Dermapterans and here around Coleopterans and Dipterans, they have nothing to do with each other. Even they belong, in these 2 cases, they even belong to 2 different types of surfaces, outgrowths. In this case, we have so-called acanthe, which are the hairs which originate from 1 single cell, and the hairs of Coleopterans, they have also the socket, which means they originate from 2 epidermal cells. So they have an independent evolutionary origin. Okay, this is an example of the smooth pad of the grasshopper. So it is, in the binocular microscope, it looks pretty smooth, but you will see later on it's not absolutely smooth. But, well, some orthopterans, they have really, really smooth pads. But if you freeze this material and using cryo-SEM and look inside of the pad in more or less fresh conditions, you see that this material is actually not a typical insect cuticle. Insect cuticle is like a layered material where the fibers, chitin fibers running in the preferred direction. Then you have sometimes the helicoidal orientation and so on. But in this case, you have fibers which are running more or less perpendicularly to the surface. They are branching in the vicinity of the surface, and the whole stuff is terminated by very thin layer of so-called epicuticle. And the gaps in between, they are filled with a fluid. So which means that you have a very soft, like ball-like structure or bladder structure supported by fibers. And you can imagine if you build contact with this kind of material, you can replicate any kind of surface. Hairy structures, here you see a couple of dipterans, airwigs, beetles. What you see are the hairs. And well, people from the nanotechnology say to make hairs is not a big deal. We can do that. But the point is that if you just make the hairs, no matter the material which you take, you don't generate strong adhesion. This is the problem. And the question is, what is different in insects? Well, we can mimic the hairs, but it doesn't work same way. Okay, because we should know a little bit more about our fly. This is the fly foot again. Well, to close adhesive pads with the hairs here. And what we should learn, and that's what we do over 15 years, you should know how the arrangement of muscle, how do they help to position the pad? What about the joints here inside? The sensors which are controlling positioning of the leg on different surfaces. We should know something about material structure and properties, because we should know which material we use for mimicking the original biological material. We should know something about the forces acting on the pad. But the most important part, and this is the part of this lecture, is the contact mechanics, what happened here at the tip of every single hair, because it is crucial for generating adhesion. And of course, we should know the potential substrates. Are they hydrophobic? Are they hydrophilic? Smooth, rough? And what about fluid? As you might know, insects also generate some fluid in the contact area. Okay, today we will focus on the contact mechanics because, in my opinion, it is the most important part of all this business. But I will show you a couple of slides about really contact. So this is again images of the fly, but let's start actually with these 3 groups of animals. Actually, we know that insects produce fluid in the contact area, which means that if the fly is running on the ceiling, it left the kind of pattern which corresponds to the pattern of the single hairs. By the way, it's a hexagonal pattern, which means that it is the highest packing density of the hair. Well, if you use a cryo-SEM, again, you see this kind of menisci, which means there is a kind of transition between the hair and the substrate. So they use kind of wet feed. How the fluid is produced and guided to the right place. So the old papers, they say, okay, the fluid is produced somewhere here above and then running through these kind of surface channels and then through the surface of all the hairs and then end up on this meniscus. If it would be true, then I'm afraid that at some point all these hairs, they will stick together if you have adhesive fluid on the surface of the hair. And this is indeed not the case. The case is different. So fluid is produced, of course, in the pad, and it is guided through the empty lumen, volume inside of the hair. And in some flies, for example, there are pores below this kind of terminal element, or terminal plate. And as soon as the fly made a contact, this little droplet of the fluid will be directly guided in the contact area. So, which means that the hairs stay clean and the fluid is produced only if the contact is necessary. Because the fluid is costly, it consists also of lipids, and this is energetical, of course, valuable kind of material. But some other hairs and some other animals, they have hairs without these kind of pores, but the lumen is going up to this, well, spatula, and the fluid can sweat through very, very thin membrane here, which is only about 50 nanometer thick. So the fluid can be just sweat off. So what animal have is a sweat feet. So in the meantime, we have also very nice chemical analysis. We know pretty much that this fluid is very complex. At the beginning, people believed that if we can make a artificial fluid, if we know the composition of the fluid, we can make it artificially. But it turns out that the fluid consists at least, just the lipid part consists at least of 30 components, which you cannot buy just using the catalogs. You have to synthesize it. So which means that the fluid is even more complex. It's not just 30 components. The fluid is usually biphasic. It means that the main droplet is actually water-based. And in this droplet, there are tiny little nanodroplets, and they are lipid-based. So this is two fluids which are not mixable. The question is what it is good for. Well, there are many different hypotheses, but still we don't know exactly. So maybe it is good to make a contact with hydrophobic and hydrophilic surfaces because fluid can wet any kind of surface. Maybe it is a good idea to use this biphasic fluid just to transport valuable little lipid droplets by using cheap water as a carrier fluid inside of tiny little capillary. So we don't know that. And another thing is that, imagine if you have droplets around 1 microns based on water immediately disappearing from the surface, but the lipid make a very thin layer. Because imagine if you have a very thick layer of lipid, then you have a lubrication situation. You don't have a sticky situation, but lubrication situation. So you should, or the animal should be able to generate very thin layer of the fluid, which is highly viscous, and do it immediately and do it very cheap. Okay, now is the question, what are indeed the basic physical forces involved in the adhesion? So you can use, there are many methods how to approach it. In this case, we use atomic force microscopy. I don't know if you're familiar with this method. Very short explanation. This is the fly. This is the fly food into the microscope. And atomic force microscope, this is in fact a mechanical kind of cantilever with tiny little needle at the tip and with a laser beam focus at the tip. So you can use a piezo drive and run a measurement in which you touch the single hairs of the fly. And at every single touch, you measure force distance curve. So you measure force you apply and the force you get when you pull off. So you measure load force and adhesion. But you can do it using this approach. You can do it not just one measurement per hair. And the hair dimension, as I mentioned, in the fly is like 1 micron in diameter. But you can, using this technique, you can measure like 30 by 30 single measurements, 900 in very quick run, and you get a map like this. So every single pixel is the value of adhesion force on the tip of single hair. What we learn from this? First of all, that in the middle of the hair, the adhesion is slightly higher. Why? Because you see there is a little depression here, and the fluid will be more collected here in the middle. And on the side, the force is a bit lower, and the force is huge between the hairs. So which means that if you get your cantilever between the hairs, you kind of stuck in between. So this probably also works if the surface is a kind of hairy or structured. But the important part in this image is that in the air, the forces are much higher. It's about 30 nanonewtons, and in the fluid, the forces are going almost to the zero, which means that in the air, we have a capillary interactions. What are capillary interactions? Capillary interactions, that's interactions. If I take the piece of paper and put it on the window, then it falls down. If I make it wet, it holds on the window, because capillary interactions are happening between fluid, air, and water interface. You should have the interface between three phases: fluid, air, and water. So, and we have this situation only here in the air. If you put everything under the fluid conditions, then the capillary forces are switched off. So, which means that the main contribution in the fly adhesion is made by actually capillary interactions. And well, this slide also actually showed more or less the same thing. But the hairs are not only occurred in flies. You can find them also in other animals. Also geckos, they have hairs for adhesion. On spiders too, and beetles too. But the hairs of the geckos, they are much smaller. The hairs of insects are much larger. Why it is like this? Look, if you look at the body mass of the average mass of the animals within the group, then you see that the density of the hairs is increasing and the size is strongly decreasing. So actually, against the expectation, the larger are the animals, the smaller are the single contacts. And we can draw the line here. These two animals groups, they use dry adhesion. They don't need any fluid, because the hairs are so thin, they can come into such an intimate contact. They can switch on van der Waals interactions, which are very, very short range forces. But insects, they actually need capillary interactions because the tip of the hairs, they're pretty large. But we see everywhere the hairs. That's why the basic physical force is the one thing, which can be different in lizards, spiders, and insects. But the contact mechanics is pretty similar. They use hairs to increase the chance to get a contact with the surface. So let's prove this kind of statement. But it's very difficult using biological means to prove this concept. So in this very moment, we actually switch to the artificial surfaces. And it's very primitive. It's, of course, not the hair, not hairs of the flies. This is just a little bumps on the silicone rubber, and we generated it just by using laser beam and drilling the hole, not drilling, but burning the holes in a piece of iron. And then we molded, we use the silicone rubber to mold this, and then we have this kind of array. But it is sufficient to prove the concept. So if you take the same material of the same size and measure adhesion, adhesive strength, adhesion per unit force area, you have much lower strength than if you have actually gaps in between. You have gaps and adhesion is higher. So which means that the contact formation is enhanced by having the situation of the hairs in comparison to the completely flat surface. The interesting thing, I would expect your question here, why after higher pressure we don't have any force? Because if you apply too strong pressure, then the hairs collapse and don't generate proper contact. So if you press too strongly, it's not good. So which means that apparently animals should control the amount of pressure, but you will learn later on it is not the case. And the problem is, well, having these kind of toys, we were able already to pick up some little tools, and already it was pretty promising and exciting. But the problem was the contact formation. We should play something like 10 minutes with the samples. And if you bring all single pins in the contact, then it works. But flies do it in 10 milliseconds. Immediately, the flies bring the food in the contact and all the hairs jump into the contact. What is different in the fly food? The difference is in the contact at the tip of the hair. Because, well, again, if we go back to biology and compare different kind of contact systems in adhesion, some of them are flat, some of them like torus-like. There are even tiny little suction cups in some aquatic animals. But the interesting result from this comparative study is that majority of animals which are able to walk on the ceiling, they have these band-like or spatula-like structures. What it is good for? Why spatula-like enable animals walking on the ceiling? Because if you use spatula, you have a very low bending stiffness of the tip, which means even if you have a nanoscale roughness which you cannot feel with your hand, for you this kind of roughness is smooth. Even at this roughness, this plate can make a contact by adapting to the surface profile. If the roughness would be stronger, would be also no problem because the hair is very long and can go deeper and still generate a contact. This is the first lesson from this image. The second lesson is, look, some animals, they have these tiny little horns, and other animals, they have little bumps or ridges here on the back side. The adhesive side is always very smooth. Why it is important to have these tiny little bumps? Because the hairs are so close to each other that without these bumps, hairs would stick together. We would have a situation, we call it condensation, hair condensation or hair conglutination. But it doesn't happen because it is a spacer. And it allows animals to use motion in the lateral direction, sliding motion to make a contact. And if you move the head, the leg in the opposite direction, then you have a debonding situation because the hair will, the tip, the spatula will flip in the opposite direction. And the opposite direction is non-contact situation. So the whole control about the adhesion is built into this little plate, which is quite clever, I would say. Yes. And you can increase adhesion without normal load. We will come later to that. Because usually, in order to use pressure-sensitive adhesives like Scotch tape, you should apply pressure. And if you apply pressure, then the substrate is working or reacting against the applied force. I will explain later on that. All the animals, also including geckos, not only insects, they have not one level of hierarchy, but several levels of hierarchy in the design, in the structure of adhesive pads. Why it is important? Because natural roughnesses, they have several overlapped roughnesses. In this room, you will not find surfaces which have one wavelength of roughness. If we take, well, the microscope and look on any surface in this room, we see first the waviness. If we take the stronger microscope, we see the micro roughness. If we take the electron microscope, we see the nano roughness. So it is many different levels of hierarchy in the roughness, which are characteristic for the natural roughnesses. So even if you cope with one type of roughness, it's not sufficient. In order to generate adhesion, you should cope with all the roughnesses. And that's why these systems are usually hierarchical. So which means that fly has pulvili. They are very flexible. They can adapt to the waviness of the roughness. Then they have tiny little hairs which adapt to the micro roughness. Then you have the spatula which adapt to the nano roughness, and then you have fluid which can cover which can cover even molecular roughness. So, in order to adapt to the natural fractal world, you need something which is the mirror of the natural fractal world. That's why these systems are so complex. And it's even more complex than just structural complexity. There is also complexity in the material composition. So here you have what we usually entomologists use, SEM image of the ladybird beetle adhesive pad. You see some, by the way, it's not very trivial. The hairs at the tip are much longer than at the base. It has some special meaning. But if you use another technique, which is confocal laser scanning microscopy, and this is a superimposed image of 3 different wavelengths of light, you learn that the base of the hairs probably made of different materials. So everything which is yellow and reddish here is cross-linked, tanned, or sclerotized cuticle. Everything which is blue, contains resin or resin-like soft gel-like materials. So you have a kind of gradient material, which is probably stiff at the base and soft at the tip. But the question is, what about the properties? Again, we use atomic force microscopy. We can go along the hair and measure properties along the hair. Because the question is, okay, what means stiffer? What means softer? It's 2 times stiffer, 5 times, 100 times. Okay, this is the fresh leg, and this is base of the leg. 1 gigapascal material properties, stiffness of the hair. It corresponds to the very stiff plastic. If you go to the tip, the property is like 1 megapascal, which correspond to the kind of hydrogel, to the pudding. The difference is 4 orders of magnitude in the stiffness. If you let it dry, then this material, if it loses water, it becomes as stiff as the base, more or less. Okay, but the question is, what is it good for? The explanation is, if you have not this gradient, if you have a homogeneous stiff material, then the tips are too stiff to build the proper contact at the nanoscale roughness. But if you would have the hair which is blue, very soft, then it would build the contact, but only once, because after the next contact, all the hairs would stick together. And the next step will be not possible. So this gradient is functionally absolutely necessary. Okay, so now I would like to ask you the question, imagine if you use classical Scotch tape. You apply certain force. If you don't apply force, you don't have a strong adhesion. So now we make an imaginary experiment. We would like to walk on the ceiling. And in order to make a contact, we should apply normal force with the first leg. What happened in this very moment, according to the third Newton's rule, all the other contacts would work against this force and try to remove you from the ceiling, right? So energetically speaking, it would be very difficult to walk on the ceiling using pressure-sensitive adhesives, because you would not be able to make a contact because the ceiling would try to remove you from the ceiling. Okay, let's ask the fly. So this is the experiment. There is a kind of special designed experiments in which we use 2 laser beams, and if we touch the leg, we can measure forces along 2 different axes. But this is not this experiment. Forget about this. This was the freely walking fly, and if the fly touch the tip, we get the information along 2 different directions. In that direction, pressure, pull off, and in this direction, 2 sliding directions. So the result is pretty surprising, because if the fly would press on the ceiling, the blue line would go up, but it doesn't go up at all. So what we see is a very strong shear force applied towards the body, and with increasing this force, also adhesion is increasing. Now you know why it is like this, because if you have a lot of spatula, if you share all the spatula in one direction, they all immediately jump into the contact. You should not apply pressure at all. And that's why flies, if they walk on the ceiling, they try to do that without applying strong pressure. Actually, you can do this kind of experiment. In order to prove this, you should not use any expensive equipment. You can just take the champion from the centrifuge experiment. These animals can generate 50 to 100 G, 50 to 100 own body weights on the ceiling. So which means if you put at least 50 other animals on this guy, they would withstand this force on the ceiling. But if you cut off 5 legs, they fall down. Actually, the simple math would tell us that it should be possible because, I don't know, 50 divided by 6 is definitely more than 1. But it doesn't work. Why? But if you left 2 legs, then it works perfectly. Of course, they cannot walk on the ceiling with 2 legs because they are not bipedal, and it occurs in other, well, controlling problems, but they can stay very long time on the ceiling. Why? Because in order to generate share, you should be able to generate counter share. So that's why you need actually 2 different directions. And if you cut legs on one body side, they try to reorient one leg from other body side and still generate shear force. Okay, maybe a little bit of contact mechanics. I think this is the only slide about a bit of contact mechanics. Why it is like this? Why we need this shear? If you take a piece of Scotch tape, you can do it at home, and put certain mass, I would say something like 100 gram, ordinary Scotch tape will withstand. But this situation is peeling angle 90 degree. My question would be, if you use 2 pieces of Scotch tape, how strongly can you increase the force? Well, you don't know, okay. Well, I expect the answer double, right? And this is true. This is true. If you have 2 pieces of Scotch tape with a 90 degree peeling angle, which you can easily do, it will be indeed doubled force. But having 2 pieces of Scotch tape, you can self-stabilize their configuration and you can reduce the peeling angle up to 30 degree. At 30 degree, you can make it 8 times stronger. With ordinary Scotch, everything the same, material is the same. It is just geometry, it is just the configuration. And this is only 2 pieces. The fly, we said, has at least 3 legs in contact, right? So which means that this effect will be much stronger if you have 3 legs. Why it is like this? Why this 30 degree is such a magic angle? Fracture mechanics tells us if you have 90 degree peeling angle, then the force you apply here will be guided to this fracture here. And it doesn't matter, do you have here 1 millimeter or 1 centimeter or 1 kilometer? The force, the force will be concentrated in this fracture line and will quickly propagate through all the line. So which means that in this configuration, the force is proportional to the width of the fracture line. But if you change the angle, then the force will be redistributed, of course, not about the entire contact, but about some area. And suddenly force start to be proportional to the area. And this is the effect of force which is used by the aim. Well, if you would like to make a biomimetics, the challenge is to integrate, as you learned from this lecture, the right dimension and the density, the aspect ratio, the slope is important, hierarchy is important, shape of the contact is important, the symmetry with proper movements during attachment, detachment, gradient materials, and some nanostructures to prevent condensation. Unfortunately, it's too complicated, so you cannot do it at once. Of course, there are some laboratory experiments, but at least you can take few of them. You can use it as a menu, as a catalog, and try at least to apply some of these features. And that was the project with a company in the southern Germany, where we try to implement what we learned from this kind of lecture into real material. And what we implemented is, first of all, the contact subdivision. And of course, we use hexagonal pattern to increase the packing density, and thin plate-like head, joint-like neck, high aspect ratio by 5. It's not very high. The animals have 30, sometimes 100. but it is a real industrial process. This is not the laboratory prototype. This is the endless production of the tape. So which means that this is actually genius because the structure have intersections and the diameter is 30 micron, which is really, really small. Okay, what about the performance? Well, flat, this was what I showed at the beginning of the talk, and this is the performance of these tapes. So if you increase the density, as you see, the performance is increasing, and you have not only stronger strength per parent contact area, which means per, well, this disc, but even per real contact area. So you have, well, in fact, the gaps are sticky. Let's put it that way. The gaps are sticky because you see stronger adhesion at lower apparent contact area. Okay. So people, of course, realize that since 2005, where the first paper was published, until now, every year, there are tons of papers, and this geometry is really, really successful. Why it is like this? Can we use this back to the biology? Can we learn something from our artificial material for biology? Okay, this is the high-speed camera experiment of single artificial hairs which have a proper structure in comparison to at least beetles. And what you see here is that if you build the contact, you are never able to make a perfectly aligned surface. You start always with one side, right? It's impossible to have it perfectly parallel. So as soon as you make a first contact, you don't have to apply any force because this thin lip, we call this process healing, not peeling, but healing. It's the opposite process. It's just run into the contact automatically. So, and that's why the contact will be formed in 4 milliseconds, just like in flight. You don't have to apply pressure. Okay, if you over compress it, if you apply too strong pressure, this tip doesn't jump out of the contact like in the previous experiment because of the lip. It's so stable that it stays in the contact. You can squeeze the material, doesn't matter. And the adhesion you measure will be similar. You have this gentle touch or you have overcompression, doesn't matter. Adhesion is perfectly the same. So which means we can speak with these kind of materials about non-pressure sensitive adhesives, just like in the fly. But the interesting thing happened if you debond, if you detach. Look, I would expect that detachment would be just opposite to this process. The void would start if you move from this direction in that direction. So the void should be started here and then peeled off. But it is not the case. What happened here, the voids start to form in the middle and propagate to the circumference of the contact. Why it is important? Because at the fracture, at the breakage, the main role is played by the fracture line. So you have much longer fracture line in this case than just the diameter. In the first case, it will be diameter of the contact. And the diameter is, of course, much shorter. The circumference is much longer. And if you multiply it by millions of contact you have, then you have, of course, much higher force. Some of you would say, okay, this is probably suction cup, because you start from the middle, the propagating, so you have a void in the middle. This is not a suction cup. If you make the experiment by structure, well, this is smooth, this is structured, and this is ambient pressure, and this is reduced pressure. There is no difference. If it would be suction cup, then it would be very strong difference, but it is not. So this is indeed van der Waals interaction plus capillary forces maybe, but definitely not suction. And what happens if you put the animals underwater? This is terrestrial lady beetle. They can walk in aquarium, no problems. If you carefully look at this video, they have a kind of silvery appearance in contact. So what we learned from this is dry contact of the ladybird beetle, and this is the contact underwater. You see what happened here. Animals stabilize air bubble underwater. they use air bubble to generate contact and still make a dry contact underwater. So in fact, they use the same mechanism by de-wetting the surface. Of course, it doesn't work for all the surfaces, but at least if the surface is hydrophobic, it works. Why hydrophobic? Why hydrophobic is important? Because the majority of plants are hydrophobic, which means after the rain, for the animal, it is absolutely no problem to make a dry contact and generate adhesion. Of course, you can do it with artificial experiment, with artificial material, same thing. Look, underwater adhesion is even stronger in comparison with the fully wetted. Fully wetted adhesion go down. In the air, adhesion is strong. With the trapped air, it's even stronger. I can explain you, but we are a little bit over the time, because here there is additional suction cup. And well, why it is like this? Why we can use capillary interactions in the air, but why we cannot use capillary interactions underwater? Theoretically, we can do it because we can generate underwater capillary interactions by putting air bubbles. But why we don't see air adhesives underwater? Because you are not able to stabilize air bubble. If you take air bubble underwater, it immediately disappears. But if you take the array of the hairs, you can easily stabilize the air bubble and use it as capillary adhesives underwater. Okay, so our material which we use, which I showed you, contaminate, and of course animals' feet are contaminating, and of course using fluid is a kind of prevention contamination, or even rinsing the tips of the hairs by the food. But what about artificial material? This is the structured version, and this is the smooth version. Smooth version contaminates much quicker than the structured version. Why that? Why? It is like this. Because if you have a structured material, the particles, they will be swallowed by the gaps. And that's why the tips are still exposed to the counterpart and still generate proper adhesion. So that's why there is a strong delay in the contamination by having this kind of configuration. But it is even more exciting because we use silicone rubbers. After 300 cycles, if the adhesion goes down, you can wash it out and the whole adhesion is coming back. You cannot do it for the Scotch tape because you change the chemistry. Here, it doesn't matter. You can reuse it many, many times. You can wash it and reuse it. Okay, so what it is good for? Of course, the company is thinking of other applications, but of course, we like the toys. And here you see the kind of robot. Okay. The experimentalist is nervous. But what is important to watch here is that it is not the Scotch tape. Look, they jump out of the contact immediately. If it would be the Scotch tape, you should generate a lot of force. So at some point, it's like 140 grams or something like this, and at some point it's falling down. It was, by the way, one of the first version of the material. So you can use it for like pick and drop. 500 cycles. Meanwhile, we know that without washing, 50,000 cycles in normal environment. But with washing, you can use it up to 2 million cycles. And from this point, if you go to the company, to other company who would like to buy it and say, okay, we have material which can withstand 2 million cycles, they say we buy it. So if you say, oh, it's working principle, too early. Okay, so we can hold a large body mass on the ceiling, if it is possible. Walking will be difficult, but walking on the wall works. And well, let's come to the conclusion. Well, I don't have to convince you that comparative approach in zoology is really one of the crucial and fundamental things. If I give this talk to the engineers and ask, would you believe that making gaps in the material would increase adhesion? They say no. They can explain it theoretically, but they would not come to this idea. So that's why the idea is originally really coming from biology. And by making these classical kind of studies we doing every day. Well, related to this talk, multiple contacts are important, terminal elements and contact formation is important. Biomimetics is promising, and we have actually two goals. If I'm honest with you, my first goal is not to make companies rich. My first goal is to understand biology better. But of course, there is some potential for industrial application. And well, today we just discussed attachment, which is our main topic in Kiel. But if you think about optics, insects, it's El Dorado. You know better than me that the coloration, the structured coloration is a great topic. And in fact, it has an endless potential for innovation. And I'm very pleased to have here a team of supporters from London South Bank University. Hamed Rajabi was my PhD student working on dragonfly flight and on the crack resistance of the wings. And now he established very, very powerful group here, not far away from Imperial on the other side of the Thames. So, and I wish you the good start. Well, 5 years already. It's not a start. It's already, everything is going. So, which means that if you have nice ideas, approach him and give him your best insects for further studies. Thank you so much.
Thank you very much indeed, Stanislav. Absolutely fascinating. I think no matter how long you've been in entomology, there are still some things that all of us sort of take for granted. And until today, for me, flies walking on the ceiling, I think, was one of them. And you now just see what an incredible evolutionary process has gone on to make that possible. It makes you wonder why flies, what the evolutionary pressures are that make flies want to walk on the ceiling in the first place, whether it's to escape natural enemies. I mean, why can't they just walk on the surface and eat our sugars and be done with it instead of evolving all those complex mechanisms for being up there? Anyway, that's a big question. Questions must be lots.
I actually can immediately answer this question. Of course, if it's a question, I absolutely agree. The enemies is one thing, but the other thing, if we look at the phylogenetic tree, we see some correlation between flight evolution and adhesion evolution.
Yeah.
So in order to start flying, animals should climb up the plants, and I think evolution of plants and flight, and I think they all go somehow in parallel.
That turned out to be a better question than I thought it would. Okay, questions? Yes, at the back.
It's amazing, really proactive. Just wondering, you studied the scalability of this kind of most technology or creating is scaling
Absolutely. Of course, all the systems in biology, they obey the scaling laws, and of course there is some limitation for the scaling law, and that's why the geckos, they have, this is probably the limit for the scaling law. Actually, many years ago, we tried to draw this line with the density and with the size of the structure just forward to the mass of the human, and we end up with something like 4 nanometers, which would be probably not a big challenge for the modern nanotechnology. But the problem is that you probably should integrate new hierarchical levels. And that's this integration would be probably difficult. So, of course, the scaling law, because the majority of you, of course, know that adhesion is scaling in the second power, and the mass is scaling in the third power. So, which means that we don't have a sufficient amount of surface to withstand larger mass. So that's why Drosophilas, they have a couple of hairs, but Calliphora, they have already millions of hairs, even if the size is not that different. So the scaling law, of course, we should, well, think about that if we integrate it. But making this structure smaller, the current technology in the big scale, in the industrial scale, is like 30 microns, it's a limit. But of course, in the lab, you can do structures which are much, much smaller. Of course, these kind of materials, they are much more effective than what I showed today, but they are limited to the, well, wafer technology, which is make them extremely expensive. These material, you can buy something like 10 meters, 400 euros, which is, if you make it tiny little stripes, and that's where company mostly use it. You can use it for medical application because it's silicone. You can use it inside the body. You can rub the wounds, for example. But Verizon in the US, they, for example, use it for adhering some devices on the walls, on the ceilings, and so. So it's going. Jeff.
So, we all have insects feeding their legs. So, there's something to do with feeding sensory organs. And you're thinking to feeding the organs instructions that are necessary for feeding. How do you know insects feeding?
Yes, we did the experiment. Well, you can do really simple experiment by putting some colorful powder or powder which is UV active, and then that's very easy. And actually, it's pretty straightforward. When I started to work experimentally on the fly using high-speed video recordings, the first thing I learned, the fly spent 80% of time by cleaning the feed. And you probably know this typical behavior of flies. Well, maybe part of this behavior has something to do with getting rid of electrostatic forces. And maybe also having this fluid is also part of getting rid of electrostatic forces. And the fluid is also, as I mentioned, important to rinse the particles out of the surface. So if you take the fly, which is contaminated, and let it start walking, all the particles will remain in the fluid, in the footprints, not on the feet. And another reason for having lipids on this system, you probably can guess if you carefully listen to the lecture. If you have resin, if you have a hydrogel, how to keep the hydrogel not evaporating, how to keep water there. You should have a coating of lipids. So that's why the function of the fluid is not just enhance capillary interactions. There are several other functions too. Sure.
That was fantastic. Oh, it was fantastic. Thank you. When plants that trap and infect the nights, mating purposes, and then go, but can't find inside. You know what to do from the insect that have a failure, they beat, more you get out of it.
It's an excellent question, which deserves another lecture. And I would be not able to answer this question, but fortunately I have Lena, who's my wife, and she is by chance botanist. And we have several projects working on really plant side of this story. And indeed, there are several ecological group of plants. They use, well, on purpose, these kind of principles to keep the insect away from the surface. Well, the first are carnivore plants. Like if you think of Nepenthes, pitcher plant, they have several tools to cheat insect. The first tool is on the rim, which is a fluid. So if you're thickening the layer of the fluid, then you have a hydroplaning, having this system. So if you have water, on the water surface, insect would, maybe on water not, but if you have it's not pure water in this case, then you have a hydroplant. The other system in the Nepenthes, they are tiny little wax crystals, which are leaf-like, and they're so tiny they can break off and contaminate the leaves. And this is the whole ecological group of plants which really use that on purpose. The another ecological group of plants are plants which use insects as pollinators, like Aristolochia. Aristolochia plants, they have a picture flowers. They can trap insect also using tiny little wax crystals which break off and contaminate the legs, and then they can open the flower next day and let them fly. It's not as nasty as the parents. And the third ecological group, which is completely overlooked, and these plants you can find even here on the meadows around this place. Many plants, they have so-called greasy pole syndrome. These plants try to keep ants away from the flowers. They have waxy stems. The rest of the plant, even leaves, they have no crystalline wax, but stem, and only flowering stem. And there are hundreds of species in our flora having this greasy pole syndrome. That's why it's, for ecologists, among entomologists, is great field.
Time for one, maybe two more front here. So, yes?
It's kind of a question. It's not be looked so much and being for me, I understand the need for the need to be able to maybe if we have a need on what's happening with the energy is done, and I actually see out of being there could be something reaction that's by a degree by how do you think of it and be by picking the series.
I completely agree. The chemical part of the story is still remain quite bad understood. I would say it works in principle even without chemistry and works well. So that's why I always in my head the chemistry is actually subordinated. It's not that important, but I agree. And we know that different insect groups, they have a different chemistry. And we wonder if the animals living in the desert maybe produce different fluid. I absolutely agree. This part is actually not studied sufficiently. So I think it's still the open field for everybody who would like to enter. I think chemistry is a great thing. And from the point of energy, it's a good point. If you mean the muscular energy, then we also know that because we did some electrophysiology on animals walking on the ceiling and animals walking on the wall and animals walking normally. Animals walking on the ceiling, they should work. They work hard. They should generate this share continuously. On the wall, it is not that critical because on the wall, gravitation helps you because the gravitation generates share. So in fact, if you kill the gecko and put that gecko on the glass, they will stay, maybe not forever, but for some time. But not on the ceiling because you need the energy. So you're right.
I think we'll leave it there because that clock is well, work our way down the road as many of us. Stanislav will be at the Verrall Supper, so if you've got any more questions, there'll be lots of opportunities, 5 or 3 down there to ask Stanislav whatever you wish to. There's one more great privilege that I have today, though. I don't know whether you know this, but the Verrall Lecture is our society's premier lecture of the year. And with that goes one of our premier medals, the President's Medal. So it's a massive privilege to be the absence of the president, to be able to present this to Stanislav. And I am told that it's made much better than the Olympic medals and won't fall apart. So, Stanislav, thank you very, very much indeed.